How is PGS performed and what does the process look like? PGS, now called pgt-a, used to be done on day 3 of embryo development, and sometimes on day 5 or 6 of development. But nowadays, the majority of IVF clinics do it only on day 5 or 6, which is the blastocyst stage. This is because unlike embryos on day 3 that have more or less 8 cells, a blastocyst is a more advanced embryo which has cells that have differentiated into 2 types of cells. The inner cell mass, that's going to be the future baby, and the trophectoderm, which is a structure composed of more or less 200 cells that will keep growing and expanding and later become the placenta. This implies that a blastocyst has a greater number of cells, so the biopsy is safer. The biopsy is performed by taking a few cells of the part that will later become the placenta, the trophectoderm. It is important to note that we do not touch the inner cell mass, which is going to develop into the future baby. After doing the biopsy procedure, we send the samples to the genetic laboratory, where they will analyze them and determine if the embryos have the correct number of copies of chromosomes or additional types of abnormalities. In the laboratory, we freeze the embryos immediately after performing the biopsy. The freezing technique we use is called vitrification. The embryos will be safely stored on our nitrogen tanks. When the genetic laboratory sends us the results, we will schedule an embryo transfer of 1 healthy embryo. If we have more than 1 normal embryo, we can also select the embryo according to its quality before vitrification and decide which one we should transfer first. We do this reviewing morphological quality during embryo development during the fresh cycle. The rest of the healthy embryos will be kept stored on our nitrogen tanks until the patient wants to have them transferred. It might sound like a complicated process, but it is a very well-studied technique which we perform often.